Myself Lokesh, I am pursuing MSc Biochemistry at JSS Academy of Higher Education and Research. Now I will be explaining about the complement system. The complement system, also known as the complement cascade, is a component of immune system which boosts the ability of antibodies and phagocytic cell to eliminate germs to and also to stimulate inflammation in organisms.
ಇದ್ಯಾಕ್ ಕ್ಲಿಪ್ ನೋಡ್ದೆ ಮಧ್ಯದಲ್ಲಿ ಇದ್ಯಾಕ್ ನೋಡ್ತಿದ್ಯಾ
Myself Lokesh, I am pursuing MSc Biochemistry at JSS Academy of Higher Education and Research. Now I will be explaining about the complement system. The complement system, also known as the complement cascade, is component of immune system which boosts the ability of antibodies and phagocytotic cell to eliminate germs and also stimulates inflammation in organisms. These components must be activated by the proenzymes that must be cleaved in order to become active enzymes. The complement activation can be started by antigen antibody complexes or by number of non-immunologic substances such as endotoxin. The complement system is triggered by three biochemical pathways: the classical pathway, the lectin pathway, and the alternative pathway. First, we will be looking about classical pathway. The classical pathway. The AG-AB complex are the main activators of this pathway, activated by the binding of AG to AB on target cell. Only antibody classes of IgM, IgG1, IgG2, and IgG3 can activate classical pathway. Activation of C1 occurs when the two globular head region of the subunit C1q binds to the Fc region simultaneously. So, one single IgM or two closely spaced IgG molecules bound to the antigen can activate C1. C1 binds to the AB in antigen antibody complex. It initiates the classical pathway and becomes enzymatically active and is referred to as C1QRS. C1 is a massive multimeric protein complex which one C1q molecule and two C1R and C1S molecule. Both C1R and C1S are proteases that helps in cleavage of C4 and C2. C4 is a protein linked to C1. C4 is cleaved into C4A and C4B. Activated C4B binds to the target surface in vicinity of C1q, where C4B cleaves C2, which is then split into C2A and C2B. When C2A binds to C4B, the resulting complex is C4B2A. The C4B2A complex is responsible for converting inactive C3 into an active form by cleaving C3A and C3B apart. C3B can interact the convertase or microbial surface. By binding to C3 convertase, the C3B simultaneously C5B to cleave the C5A and C5B by enzyme C5 convertase. After the binding of C5B is held in the place by C6, whereas C5A is free and to diffuse away. After the binding of C8, the C5B C7 C6 complex is introduced into the phospholipid bilayer of the cell membrane. All these are activated proteins in the complex create holes in the bacterial cell wall which causes water and salt to diffuse into the bacterium through the hole and the bacterial cell wall cells and eventually burst out. So this is all about classical pathway. Next we will be looking about alternative pathway. Upon encountering foreign surface, the 3B protein binds directly to this surface and unlikely the classical pathway does not require the formation of antigen-antibody complex. The hydrolyzed domain of C3 undergoes structural changes exposing the binding site for factor B. Mg2 plus is the only ion required for functional activation of the alternative pathway that stabilizes the interaction between C3b and factor B. This C3 bound factor B is cleaved by serine proteins factor D forming C3BB. CB convertase. C3 convertase in the alternative pathway is unique as it is made up of subunit C3b and BB. The convertase complex in turn cleaves C3 and to C3a and C3b. The C3a fragment is cleaved into the fluid phase when a second C3b adds to it. The C5 convertase is synthesized. C5 convertase cleaves C5 into subunit C5a and C5b. C5b reunit C6, C7, C8 and C9 forming MAC that includes cell lysis. Next, we will be looking upon lectin pathway. Mannose binding lectin (MBL) is structurally similar to C1q in the classical pathway and allows binding on many pathogens. The recognition of mannose containing sugar on pathogens by MBL initiates a reaction, like C1q in the classical pathway. MBL is a six-headed molecule that forms a complex with two protease enzymes MASP1 and MASP2. When the MBL complex binds to the pathogen surface, MAS1 and MAS2 get activated when cleaves C4 and C2. C4 is cleaved into C4A and C4B. C4b attaches to the surface of the pathogens including C2 to bind, which is cleaved by MASP2 to form C2b and C2a. C2a binds to C4b forming complex C3 convertase. The convertase complex in turn cleaves C3 to C3a and C3b. The C the 3A fragment is released into the fluid phase. This complex C4b2a3b, the C5 convertase synthesis proceeds on the surface membrane of the pathogen C5 convertase, cleaves C5 into subunit C5a and C5b. C5b reunit C6, C7, C8 and C9 forming MAC that induces cell lysis. Next, we will be looking on some biological effects of complements. Cell lysis. The most important purpose of complement activation is to lyse the microbes that have entered into the host. Complement activation leads to the lysis of bacterial, fungal, viral, protozoal and many other cells that the membrane attack complexes. Inflammation. During complement activation, some of the components are split into complemental fragments. The large fragments continue the complex cascade, while the smaller fragments play important role in inflammation. Thank you.